All right. Good morning, everyone. Welcome to our first Biotech Connector Speaker Series for 2026. My name is Vladimir Popov, and I'm the Chief Innovation Officer here at the Frederick National Lab for Cancer Research. I want to welcome all of you folks that are here in person. Please get some coffee and breakfast. And I also want to welcome people that are online. I think we have quite a few. A little bit of a background for those who are here for the first time. The Biotech Connector event is a joint initiative between Frederick National Lab and the Frederick County Chamber of Commerce. This event celebrates and strengthens the Frederick County biotech and bioscience ecosystem. It showcases recent local and kind of regional innovations, if you will, and creates valuable networking opportunities for scientists and entrepreneurs and business development folks. With Biotech Connector spotlighting local biotech progress and networking, I do want to take a moment and mention a very important event that's going to be happening in a couple of months. And this is our biannual event that we do jointly with Hood College. So I would like to just point out that our Reproducibility in Science Symposium is happening in June 16 through 18 this year at Hood College. So pull your cell phones out, scan the QR code. I believe registration is open. So as of a few days ago. So please check it out. We have very good distinguished speakers from across the country. And we there are somewhat of limited spots, not to pressure you, but it's been a great symposium over the years. This is the fourth one that's happening, and it's been growing. So please check it out and attend if you can. Now, let me introduce our three distinguished speakers who will discuss advancements in drug development using AI and computational science. First one is Trent Balius. He is a computational scientist at the Center Cancer Research Technology Program. I'm sorry, my coffee is not fully finished here. At CRTP, here at Frederick National Lab for Cancer Research, where he develops and uses computational methods to improve therapeutics focusing on RAS cancer targets. Trent graduated from University of Pittsburgh at Greensboro with a bachelor's degree in applied mathematics in 2006. In 2012, he received his Ph.D. from Stony Brook University, where he worked in Dr. Robert Rizzo Group. Trent pursued a postdoctoral work with Brian Shokat at University of California, San Francisco, and for a year at University of Toronto. An interesting fact about Trent, as an undergraduate, he studied abroad in Granada, Spain, for 9 months and studied Spanish language and its culture. That is impressive. Our second speaker, Abhishek Kognole, is a scientific and business development manager at SilcsBio. Abhishek received a bachelor's degree in chemical engineering from University of Mumbai and a PhD in chemical engineering from University of Kentucky, where he was focusing on application of computational approaches to study protein-ligand interaction. During his postdoctoral studies at the University of Maryland, Baltimore, Abhishek worked at the computer-aided drug design. He brings extensive experience in molecular modeling, enhanced sampling, and force field development at SilcsBio. He's passionate about integrating computational approaches with experiments for faster and better drug design. Welcome, Abhishek. And last but not least, our third speaker, Simon Wang, is an associate professor of medicinal chemistry at Howard University College of Pharmacy. And he's also director of AI and Drug Discovery Core for the District of Columbia Center for AIDS Research. Dr. Wang earned his PhD in computational chemistry from University of Florida and completed his postdoctoral training at Cornell University. With over 25 years in AI-driven drug discovery, computational biomedicine, and biomolecular simulation, Dr. Wang advances practical applications of AI for therapeutic innovation. He has authored nearly 100 scholarly works and delivered over 250 presentations and invited talks worldwide. Dr. Wang also serves as a reviewer for a variety of journals, and within NIH, NSF, and NASA panels, and he has consulted for Federal Reserve Board and evaluated projects for regenerative science talent search. Outside of work, Dr. Wang is an avid pickleball player, and he's currently aiming to break into the 4.0 level, which I understand is quite challenged. So great job, Dr. Wang. And now let me invite our first speaker, Dr. Trent Balius.
Thanks for the introduction. So as Vladimir mentioned, I'm Trent Balius. I work as part of the NCI RAS initiative leading the computational chemistry efforts. In today's talk, I will um, describe what we do in the RAS Comp Chem team. I'll introduce you to large-scale docking screens, virtual screens using UCSF dock. I'll describe I'll briefly describe two advancements we've implemented into UCSF dock and discuss an example from the literature of comparing docking with a machine learning method. So I just want to acknowledge the people in the RASComp Chem team. Everybody at the NCI RAS initiative. It's a collaborative place. The doc community and our computing resources here. So I just want to say that we're focused on RAS at the RAS initiative because it's such an important cancer target. To be conservative, it's RAS mutations drive over 20% of human cancers, and for specific types of cancers like pancreatic cancer, it's 95% of pancreatic cancers are driven by mutations in KRAS. In the RAS Comp Chem team, our main objective is drug discovery. We use molecular docking, chemical informatics, molecular dynamics, and more and more now machine learning to impact our drug discovery efforts. Our main tool is molecular docking, and we use these other methods mostly to support our docking calculations. We're one of the groups that develop UCSF doc. One of the main applications in our group is large-scale virtual screens. We also can predict binding modes and perform analog or identify analogs for known binders. So for using doc as a ligand discovery tool, the way we do this is we have a virtual library of hundreds of millions of small molecules, and on our computer cluster, we'll take each molecule and dock it into a site of interest on our protein, assign a score, and we'll get a rank ordered list of these molecules, and then we'll purchase molecules at the top of the list for experimental testing. A thing that the community is struggling with right now is the growth of chemical space. So it's growing pretty exponentially. Companies are willing to sell these make-on-demand molecules. So they're on the Zinc database, which is the public database that we use. There are now over 10 billion small molecules that are prepared for docking, and there are over 100 there's about 100 billion molecules in the database, structures, chemicals that companies are willing to sell, make and sell us. There are many examples in the literature of doc success. Here's well, I guess you can't see my pointer, but anyway, the top citation is a recent review. And then I provide a couple other examples from the literature. From my own work, here are two examples where we deployed large-scale docking against two targets. This is from several years ago now, and we found potent and selective small molecules. Here's one. I'm just focusing on one of those literature examples. Oh, my pointer is there. What do you know? This is from the Scheuket lab, where they docked to a transmembrane protein, and they performed two screens. One, a smaller screen of 2.7 million molecules, and then a screen of 1.2 billion. And then they compared the hit rates. So for the smaller screen, they had a hit rate of, my apologies, a hit rate of 13% of, well, for the larger screen, it was 36%. And then also they've discovered more potent molecules from the bigger screen. And so this is just confirming that bigger is better. And this isn't the only example. There are a few examples now from the Scheuket lab that kind of confirm this. So UCSF Doc is the oldest Doc software. It was developed at UCSF and Tak Kuntz's group in the 80s. It was originally written in Fortran, and then it was translated into C and then C++ around the turn of the century. In Doc, and when it was translated into C, this algorithm called anchor and grow was implemented to account for the flexibility of the small molecule. So, to explore the conformational degrees of freedom of the small molecule. When Brian Scheuket started his group, he was a graduate student in Tak's group. He continued to develop the Fortran version of Doc, and he implemented this method called like a hierarchical traversal method, where the ligands degrees of freedom were explored outside of Doc, and then you just traverse through these hierarchies to explore the ligands degrees of freedom. And I'll discuss that a little bit more later. Doc, so both versions of Doc are currently being developed. When I came here, I ported the hierarchical database method into Doc 6. I did this because I find Doc 6 easier to develop and maintain. Also, the hierarchical database method is much faster than the on-the-fly anchor and grow method. With anchor and grow, we're spending about a minute per molecule, while hierarchical database, we spend a fraction of a second per molecule. And it's actually this method that enabled large-scale docking. So here are just two papers from the RasCompChem team where we discuss some of our improvements in doc development. In this first one, we talk about porting the HDV method into Doc 6. And so as I mentioned, in HDV, we identify the ligand degrees of freedom, identify rigid segments, and then we do a conformational expansion where we get an ensemble of conformations about about one of these rigid rings, and we can do this for a second ring. So you can see how the conformational ensemble looks. We also pre-compute energetics, so we identify the binding site of interest, defining it by spheres, and then we'll pre-compute energies on a grid about those spheres. And then we take each rigid segment, we orient it onto these spheres, we traverse through the hierarchy, and we obtain a docked pose. Here I'm showing an overlay of an experimental pose with a docked pose. So to evaluate this, we performed an illustrative virtual screen where we, on the sauce pocket of RAS, we docked 23 million fragment molecules. We did this with both Doc 3 and Doc 6, and we can compare. I also wanted to mention, we also did, for the top 50,000 molecules, we redocked them. And then we also did a torsion minimization, and these two steps were easier in Doc 6. In fact, the torsion minimization is only possible in Doc 6. We compared the energy distributions between dock 3 and dock 6, and we see very similar energies. When we redock with increased sampling, our energies actually shift lower, indicating that we're finding better scores. So, and then with the torsion minimization, it shifts even lower because we're allowing the ligand to adjust to the protein's environment. We can look at the timing. So dock 6, it is slow. It was slower than dock 3 in this publication. The other interesting thing is increasing sampling and performing torsion minimization shifts the charge distribution of our top hits. So by doing this, we'll find different molecules, prioritize different molecules. So in this case, shifting from molecules with +1 to molecules with +2. I don't know if this is a good thing. It's just indicating that we're finding different molecules. Now with some code optimization, dock 6 is faster than dock 3.7. I don't really have time to discuss the covalent docking method. I just want to say that with covalent docking, we're performing for covalent drugs. There's chemistry that happens in the binding site. So where you actually form a covalent bond with non-covalent drugs, it's driven by through space interactions. There's no chemistry happening in the binding site, but with covalent drugs, there's a reaction, and this allows us we can take advantage of this to limit our degrees of freedom. And so we just explored the degrees of freedom around this bond, and so we've implemented an algorithm called anchor and grow. I'll skip this slide. So machine learning methods have a lot of advantages over traditional docking methods. I'm just going to focus on one set of machine learning methods around this protein folding problem. So a few years ago, AlphaFold came out. It got a lot of attention, and many people say that AlphaFold has solved the protein folding problem. And the way it works is you give AlphaFold a sequence, and it predicts the structure. Further developments of these methods have allowed for co-folding methods, and in these extensions, you can give a small molecule and your protein sequence, and then it will predict a complex. And this is actually, so it's analogous to the docking problem. And the biggest advantages of this is that you need less expert knowledge to perform these calculations. You don't need a structure or a model to perform them. You just need the sequence. And receptor flexibility is inherent in these methods because you're folding the protein around the small molecule and predicting how the molecule binds. So you also don't need to know where the molecule binds. There are some weaknesses, though, and that's really the cost and computational expense. So these methods they run on GPUs, and they take minutes to perform the calculation. The other deficit is that there's a lot of memorization in these methods. So if you're trying to predict something new, something that hasn't been seen before, they're not going to be very accurate. So this is an example from from Jin-Kun Liu's lab at Rockefeller, where he compared docking 700,000 small molecules to a GPCR or to a transmembrane protein with dock 3 and with AlphaFold 3. And he bought 100 molecules from each screen and tested them. And surprisingly, Doc actually had a higher hit rate. It was 2 times more, um, and also the expense of alpha. So Doc had a better hit rate, and it's so much cheaper to do the docking. And remember, also bigger is better. So here they performed a very small screen to compare the two methods. They cannot access that space very easily currently with co-folding methods. So um, I see Doc as still state of the art. That it machine learning methods, I see them as complementing docking in some of these virtual screening efforts. So in conclusion, I told you about the work we do in the RAS-CompKim team. I described this hierarchical database traversal method we implemented into Doc. I described a illustrative virtual screen. I introduced you to this covalent algorithm we implemented, and I described this this comparison of co-folding with docking. And with that, I'm happy to answer questions. Oh, maybe you can go first, and then Andrew. Um,
The method that you listed that showed DOC3 is faster was a point in time. Is there a horizontal approach where you could align better results with AI if we're done across more molecules at once in a greater volume?
there are lots of ways I could imagine to speed up some of these AI methods. I don't think you're going to be able to get the speed at which we're doing docking with these co-folding methods. Not to say there aren't other AI methods. There are lots of things out there, like ways to accelerate traditional docking methods using AI, and I think those are probably the way to go. Or also including these methods at the beginning or the end of our calculations. So you could imagine using co-folding to predict structures, and then use that structure to perform the virtual screen. And then at the end, you could rescore the virtual screening results with co-folding. That's how I see integrating some of these methods into our workflows. But it's a good idea to uh, group these calculations together, and people have actually deployed that in traditional docking methods. Uh, go ahead, Andrew.
Yeah, I'm gonna ask a similar question. I'll ask a different question since you already addressed that one. So on the docking you've done, the number of compounds that looked like they were actually tested were very small compared to the library. So the example you gave where they've done this for the millions versus the billion, it was looked like it was less than 100 compounds were actually picked to be tested in reality. And is that because they are so well distinguished in the scoring that it's obvious, or do you just say, well, this is the list, and I can afford to buy 100 compounds, and I'll just take the top 100.
So hit picking is an art form in docking. The last filter is always the computational chemist, where we'll actually go and sit in a room together and we'll score the molecules ourselves. As Brian Schoikert used to say, we want to win. So we're not wanting just to use the computer, have the computer tell us what to buy. We want to believe in the molecules we buy. We want to buy molecules that are going to work. So we always do a hit picking step. We'll tend to visualize the top scoring molecules, whether it's the top 5,000, but with smaller screens, you're looking at like a bigger percentage of your hit list. As you go bigger and bigger, you can't continue to look at the same proportion of your hit list. And so it's definitely a concern. And that's another area where machine learning could help, is maybe as a step where you can pull up the molecules that you actually care about from, a comparable size of your list. But yeah, hit picking is kind of an art form. I guess how many? Oh, maybe Tommy, and it looks like another question.
So when you referred at the beginning to the chemical space increasing, how significant is that chemical space in terms of its complexity? Like, is it really exploring the chemical space in a dramatic way with stereoisomers and and different ring structures, or is it just sort of incremental and not really that? Like I think of natural products and their complexity compared to some of the synthetic molecules. People don't want to make natural products because they're hard. But then on the other hand, that's chemical space.
So in that original paper, from my work, the two examples, that Nature paper, we discuss this growth in chemical space and asked whether more clusters, like more, is it more chemically diverse? Are you adding both within clusters, or are you adding new clusters to chemical space? And both were happening. So you were expanding within a cluster, but you were also growing the number of clusters. And so the space was growing more diverse, and the space was increasing the size. And we actually quantify how much. I can't really remember the numbers, but it was like both things were happening. So we weren't not every new molecule was increasing, was creating a new chemotype, but you were adding new chemotypes and expanding existing ones. And I think that's continued. And there's also there is a growth of there's this idea of what's the word, bio-like molecules versus synthetic-like molecules, and trying to explore those in this ever increasing chemical space. And I guess there was one more question, or we don't have time.
Yeah, after that presentation. Yeah.
Okay. Thanks.
Okay. Thank you. Thank you, Luba, for inviting me here. And thanks, everybody. So my name is Abhishek. I'm an application scientist and scientific and business development manager at SilcsBio. I'll be talking about our unique method that allows overcoming the trade-off between speed and accuracy in the computer-aided drug design. All right, thanks. Yeah, thanks, Trent, for setting the stage there. But in computer-aided drug design, the relative binding free energies are kind of the lead the way, and docking is one way to go from billions of molecules to hundreds or maybe thousands of molecules. But then down the stream, you always take into consideration more enhanced sampling of the the protein ligand complex to understand which molecule is binding the protein molecule with more affinity. And in this case, the traditional methods that have been there in the field are free energy perturbation and thermodynamic integration, which both require an ensemble of conformations for each ligand modifications that you're trying to build. So every time you change a molecule, and you have to start with a good starting structure, reliable force fields, good molecular dynamics, and free energy perturbation software, lots of cloud computing or HPC resources, and then at the end you get that one relative binding free energy, whether the modification is favorable or unfavorable, right? So in a given campaign of drug design, you have to do this changing the molecule every time, and you will have to climb that hill every time, right? So our co-founders Alex Mackerel and Olgun Guvench at the time were like, this is ridiculous, there must be another way. So what if we come up with a method that gets us both qualitative information on the target and as well as the quantitative information for the ligands and their ability, and you don't have to run this big calculation of protein ligand complex again and again, right? So that's how the idea of Silks came in, and the idea is that we do that one big one-time computation of ensemble of this protein with small probe molecules that are representing different functional groups and water, and we generate this entity called frag maps that is encompassing all that protein wants to interact with, and we can use that for all the downstream calculations, such as estimating ligand binding affinities and high throughput virtual screening and ligand optimizations, right? So let me introduce the method. So SILCS, it stands for site identification by ligand competitive saturation. It's a simple fundamental idea that you put your protein molecule in a box with these chemical probes, small probes that are representing different functional groups, and along with water as well, they're competing with each other to occupy these different sites on the protein. And then we extract this information and analyze them to process and generate these pre-computed ensembles called affinity maps that can be used downstream. So how we do that? It's one big computation for the target where we run almost 1 microsecond of molecular dynamic simulations, which are divided into 10 independent runs of these molecular dynamics and grand canonical Monte Carlo simulations. So the molecular dynamic simulation, so note that there is no ligand molecule. It's just the protein molecule and these small probe-like molecules that are competing with each other. And we run this molecular dynamics and grand canonical Monte Carlo. The molecular dynamics allows us to sample the flexibility of the protein as well as the interactions of these probe molecules. And the grand canonical Monte Carlo allows us to insert, delete, translate, rotate these small probe molecules into the deep pockets of the protein that allows us to get over that rate limiting step of running long MD simulations to achieve these kind of samplings, right? So this is an enhanced sampling, and how we extract the information is we divide the space of the box into these 1 angstrom voxels and find out where exactly each probe molecule has sampled how many times, right? So if you look at the representation, you'll see that when you don't have the protein molecule there will be a homogeneous distribution of these probes in the box. But when you have the protein in the box, you'll see some of the probes are interacting more favorably, meaning that their probabilities have increased around the protein, and there are also going to be lower probabilities compared to the bulk probabilities. And then you have regions where there is no probability, meaning that the protein was occupying the site. So we use that information and Boltzmann transform it into these grid-free energies that we call frag maps. So you can visualize these frag maps in the perspective with your original starting structure, where you will be able to visualize all these frag maps that are representing apolar or hydrophobic regions, hydrogen bond donors, acceptors, and charged regions. And you can additionally use different GFE levels that allow for the regions that will bind the specific chemical class more tightly to be identified. And then when you look at the, typically you visualize the protein with this surface representation, right? But the part where these probe molecules were not able to sample is actually a more underlying surface, which we call exclusion map. So you can see the difference between the surface and the exclusion map, which allows you to look at these deeply buried pockets that are typically not able to be identified, right? So this is the only region that your protein didn't allow for these probe molecules to sample. So this is really important in information that we get out of SILCs. And then how do you use this information of frag maps to score the ligands, right? Now you can we can take the ligand pose and overlay in the field of frag maps, and we can then take each ligand atom and assign a frag map type to it, right? And then based on the location of the atom in the and the grid free energy at that location, that then the atom gets the contribution, and then you can get the total ligands favorability by summing up all that atomic contributions into what we call as ligand grid free energy. And this is a representation of your ligand binding affinity score. Okay, so now we can also subject the ligand into a conformational sampling using what we call SilcS Monte Carlo or SilcsMC docking. It's not the similar docking that Trent was presenting, where you need the protein molecule to be there, but what we are docking is in the field of these frag maps. So technically there is no protein atoms there, it's just the field of these frag maps and that exclusion map, which allows us to get away from forbidden region. So we can sample the translational, rotational, and torsional space of your small molecule in these frag maps with the help of our small molecular force field generator. You can also get away from bad conformations through the intramolecular energy. And then finally, you get a pose that gives you the best LGFE score. And we have validated this score of ligand grid free energy against the state of art free energy perturbation methods that are out there. And just right out of the SilcS calculation, you get really reasonable delta G values. And then let's say you're doing this iteratively in ligand drug design campaign, and you start generating experimental data as well. You can use that experimental data to further reweight these frag maps, and what we call Bayesian machine learning, and that allows you to further improve the predictability of your binding affinities using this method. So we have shown that this is highly and on par with the state-of-the-art free energy methods. And the difference comes in when you look at how you can scale this in a drug design campaign. Said that the free energy perturbation, if you're running it on small number of molecules, it will take you, per ligand, it will take some time. But as you go into, hundreds and thousands of ligands, that time is just going to increase linearly, versus for SilcS, you run that one frag map calculation for your protein once, right? And then all that ligand binding affinity calculation is just seconds of GPU time. So in a traditional small molecule drug design approach, the SilcS methodology can be used by starting with a crystal structure of your protein, or a homology model, or an AlphaFold structure, and then you generate the frag maps first, and then you can take that frag map and use our hotspots calculation, where we basically do SilcsMC sampling of small fragment-like molecules, which are larger than our 8 probe molecules that we used in map generation to get their poses and then do a clustering to get these hotspots, we can directly use the frag maps themselves to generate pharmacophore hypothesis at a given hotspot, and then we run a pharmacophore screening, which is much more faster than a traditional docking as well. That you can then take those top hits from pharmacophore screening and then run our pose refinement calculations for generating the ligand grid free energies, and then you can go into the loop of take the top molecules, go through the experiments, take the experimental data and run these Bayesian machine learning and ligand optimization to generate more and more ideas, right? So this approach has been validated with recent example here showing for a beta-2 adrenergic receptor by our co-founder Alex Mackerel, where they started with a intermediate structure for an inactive and active from the of the beta-2 adrenergic receptor, and they generated the frag maps, generated the hotspots, and chose an allosteric binding pocket from those hotspots, right? And you can take this site and then build pharmacophore hypothesis in that binding site, and they use these two ready to order databases and ran those through this pharmacophore screening, take the top hits from each pharmacophore hypothesis, run our SilcsMC pose refinement to get the LGFE scores, take the top 1000 molecules of that, run further clustering and filtering, and then finally as Trent mentioned, hit picking to take some compounds into the experiments, and then they found both positive and negative modulators for B2AR. So the key advantages from this approach begin by like doing that one big calculation upfront, right? And you are accounting all the explicitly accounting for the protein flexibility. So you can see here that how a side chain, if it is moving during your MD simulation, that will not be part of the exclusion map, meaning that the fragments we are able to sample those regions kind of thing. And then you're explicitly accounting for solvation effects because water is also one of the competing probes, right? So you can there are frag maps for water, so you can use those to learn about crystallographic waters or the waters that can be replaced in a ligand design, right? And we can support membrane proteins and RNA in the same using the same method. So overall, the same method has been taken forward by our co-founder in applying it for covalent docking. You can use the same frag maps for one protein and run the frag maps for another protein, and we leverage that in calculating protein-protein interactions as well. And we provide that in terms of learning about your biologics development. And yeah, as I said, you can use Silks water, which is, you can learn about where water can be docked or like modeled or displaced. So yeah, overall, that's it. Thanks for listening, and I'm happy to answer any questions.
So could you imagine using, or have you used this method to identify glue-like compounds between two proteins?
Yes, there are currently ongoing efforts to use the same method for both molecular glues as well as PROTACs, right? Where you can look at the linker molecules as well. It is still a difficult process because we need to run the frag maps for the two molecules, and then when you run the two frag map generation with two molecules attached together versus them separately, you're going to see some differences which can be used in both ways, positively. But yeah, we have recently published a paper to apply it for PROTAC molecules, but exactly molecular glues, it's still ongoing.
I had a question about the, I guess, kind of practical limits that you think exist on the size of the protein. I mean, those proteins look relatively small and relatively simple, so they're single domain and probably not very big. How big do you think you can go before it becomes non-practical, or how a protein with a lot of flexibility? I guess complication is more complex.
Yeah, that's a very good question, and we have looked at, so started with the globular proteins, small proteins. We have used the same approach for membrane proteins, so GPCRs or transporters, and then ion channels as well. So it is applicable to bigger and bigger molecules. And we have used this approach for biologics, where you're looking at a map, where you don't have to run the good thing is that you don't have to run the big protein all together. If you have different domains, you can run them by separating it, and then you can basically translate your frag maps by aligning to a bigger protein where an assembly of different domains comes together. Yeah.
So as teams start to deploy like multiple Silk modules across like different programs, how do you manage like computational artifact provenance between those programs so they can look at targeted results and comparability of predictive factors?
So what you're trying to ask is, like, when we incorporate this Silk's approach with other tools and methods that you need in drug design. Yeah, so Silks basically comes really early stage, right? So typically when you're working on drug design campaign, you're not changing your target every day, right? You're deciding on your target. So the main help that Silks brings in is, like, you give your target PDB to your computational chemist. He runs this one calculation. You can distribute it to all your medicinal chemists and computational chemists, and they have that throughout the campaign of your drug design, which allows to go back and forth and discuss things. So if during the process the protein gets developed further, you can run the simulations again. But yeah, it's very early stage calculation. So yeah, I don't know. We can talk about more. Yeah. Okay. Where? Which one? Oh, okay. Right here, right?
If you need laser, you can use just use the mouse.
Oh, okay. Okay.
This one, because that one, we have virtual attendee, so they won't see it. So maybe use this.
Oh, okay, okay. All right, no problem, no problem. All right, hello everyone. So yes, yeah, thanks for the invitation and also the very nice introduction. So yeah, my name is Simon. Simon Wang came from Howard. So I would say another side of the DMV region, right? Yeah. So yeah, my lab also serves as the core lab. We provide the service to the DCCFAR. DCCFAR also is funded by NIH, the anti-HIV studies. All right. Yeah. the title is about the AIDD. So AIDD, I mean, there are different way to spell, right? So now I think the most popular one is the AI-driven drug discovery. That's the most popular the spelling. So we have to talk about the very recent the collaboration is a partnership between the Eli Lilly and the Nevada, right? Nevada. I think so they negotiate starting from the October. Now then they signed the deal is just last month in January. So it's $1 billion deal. So they can build a very large the computational center using the framework from the Nevada, and also the Nevada already have the platform by NEMA. So this kind of things is happening quite a lot actually in recent years, in recent like 2 or 3 years. So the big pharma, they really want to just like have very heavy investment. So on the AIDD. So it's different. And I'll say that. So I'll talk about later. It's different from I'll say we have the AIDD came out probably around 1915, right? So that 1915 to 1920. So that probably that is the very first the peak time. I think now is the second peak time. So so we can talk about that. So the reason why, so why now they are getting more and more serious about AIDD is because the drug discovery effort. We all know that, right? So it's very risky business. So it takes a long time, and it costs a lot of money. Normally it's $1 billion per drug, and also the risk, the chance is very low. The risk is very high. So they overall 95%. So it'll fail eventually. So that's the sense. But I'll say that so since 2015, right? So I think the number is they're over 180 or over 200. The AIDD company or so the form at that time. So they have a lot of the pipeline. They develop a lot of pipeline, and so many of them already like they go to the phase two. Goes to the phase 2, and some of them has go to the phase 2. So that's the data just taken from all the pipeline. So it shows it really work. It really work. So AIDD it can really can cut the time. The very first one, they cut the time. They cut the time by at least 50% to 70%. It's mainly this part is mainly for. This part is mainly for the preclinical part, right? Preclinical part we know that is normally is like 3 to 5 years. Now there are many cases. It's just 1 to 2 years from the preclinical part. Yeah, it's very impressive. It's very impressive. So the cost is because we reduce the time, right? So the cost is like is 30 to 60%. And also the success rate is increasing actually especially you have the data for the phase 1, right? Phase 1. So the success rate is increased to the 80 to 90% from the 40 to 65%. So I'll say that is very promising. It's very promising, and I mean that's the very recent. So even OpenAI. So they try to so just get to the AIDD business, right? So that's very recent. The Bloomberg report. So yeah. So this is the map. Right, it's from the I'll say that's the very like the first peak time the AIDD company formed, right? So around like the 2020. You can see at that time some the big pharma they just try to partner with the small, the very specialized the AIDD company, right? Like the Sanofi, the partner with Excentia, and the Lilly with the Atomwise, right? Atomwise is in US. So yeah, so that's the so that is a trend. So so it's back to the 2020, right? But yeah, yeah. So the yeah, let's go back to the see what is the AI, right? Since we call it the AIDD, AI. So the artificial intelligence. So that word came out as very long time ago, right? It is from the very famous Dartmouth conference, right? But after that, because of the hardware and the problem, right? So we call that is it's very like AI, the hibernation, the period, right? So there's no development, but after I think right around around 2000, right? 2000. So the deep learning, they came out. Yeah, so the algorithm get fully developed, especially for CNN, right? The convolutional neural network. So you can see that so it really so the power of the AI is really it's getting much stronger. So it's in the game field, right? They can very it's a very impressive performance. And also now is the so this kind of AI and the model building process is become like this very like it's daily activity for us, right? So whenever we log in, so you can just like it's a model prediction, right? It's model prediction. So yeah, the AIDD it cover is not only for the drug discovery, it can cover still a lot for it could be for clinical trial design, right? Also, it can be a part of the manufacture and the pharmaceutical manufacturing, right? And so the algorithm behind that. So here I would say the AI, especially when we talk about AI, mainly, right? Mainly we talk about its deep learning part. I would say it is the deep learning part. Right, so so we have the perception, we have the RAN, we have the CNN, right? So and the scheme on the right side mainly is for the machine learning, is for machine learning. It's regular machine learning, We have the KN and we have SVN. Right, and so since we run out of time, so yeah, I'll go through that very quickly. So yeah, so it is like the so machine learning also it is very at the interface of the statistics and also the computer science, right? It's a different problem. So yeah, so I happened to work on that. So on the machine learning, it's quite a long time ago. So yeah, so this is very first thing. So we build up. It's also is molecular docking problem, but we're using the machine learning to solve the problem. So it's also about the binding post. But we build up the model. We build the machine learning model to distinguish so relative pose and a non-relative pose. And we combine the machine learning model to the regular scoring function. It really enhance. You can see that the green bar, and it is a plus. It is the machine learning model plus the regular scoring function. And then the black bar is just a regular scoring function. So the performance is really enhanced. So that's the year of the 2012, right? And it's the very first paper we published. And also there are many like the docking competition, right? So yeah, we participated docking the competition, and we score well, yeah. And later we think about that. So so how about we can apply to more like the general docking problem. We know that normally, right? Normally, so when the drug target came out, it has multiple the structure. It's not just one or two. For example, some kinases can have over like 100, 200 like crystal structure, right? So yeah, so some of normally the docking things, the screening things is just using one or two, right? Mainly just one or two, so the crystal structure. But then think why now? So we can utilize all the available crystal structure, right? So in that way, the chance to get a diversify, the ligand will be very high, right? Very high because the different structure they're holding the different ligand, right? The different structures, the ligand. So yeah, we build up this kind of the ensemble learning models. So for individual, so each individual the quick structure is just they will build up one model make the prediction, and then we're using the meta learner and for learning process, and the right, and then so the performance is the performance is very good. Actually, it showed that so this kind of so this kind of the ensemble learning is it really can outperformance the single structure based the docking result, right? And then we apply that. So later we apply that to some real cases. So this is like the non-alcoholic the fatty acid disease, right? So yeah, we're using the human FSR the target. Then we identify as very good the ligand. So so you can see the red one is the new ligand we identify. So the serum and the pink one, it is one so the known ligand, right? So the ligand we identify using this measure is very comparable with affinity, is even better of affinity, and also we identify additional hydrogen bond with the tyrosine 350. So yeah, that's the thing. We know that, right? So if you have additional hydrogen bond, that means a lot to medicinal chemist, right? So it can increase the potency quite a lot, right? So it's published in the cover paper for JCAN. And also we apply that for the PGK1, the target. So and we did identify the 13 so the good ligand out of that. So the PGK1 is a very good target for the anti-cancer, right? Anti-cancer also for neuroprotection. And HDAC3. So yeah, so we identify the so one good ligand. So they're using this method, and then we design so the similar analogs out of that ligand. So this is the docking result. Yeah, so it's still in process. Yeah, some of the publications in all time, right? So you can see that from timeline. So we're working on this kind of the AI, the machine learning. So the based drug discovery, quite a long time ago. Yeah. So and now, so with the large language model and the generative AI, so we're thinking why now? So we can try using generative AI, right? Generative AI. So since it's so powerful, so we're using the method we call as the in-text learning. So basically, it's still prompt based. So you just tell the language model what kind of ligand you want. You can just see you can specify it. So all the chemical, physical property, also the biological, all the properties, right? You can see so the one so in the middle column, so there is a molecule we generated, right, using the large language model, and then the one on the right side, the right column is it is a target. It is a target molecule. So you can see, by using this in-text learning, so the large language model can generate is very close, all exactly the same, the molecules. So you're using this method, and yeah, we can use that for drug discovery, but also we can use that for like library design. We know that, right? So we can build up the Fox library for kinase or for GPCR, right? Using this method, using the generative AI. So right, so we fine tuning. We fine tuning a lot of the large language model just for that purpose. We're using the GPT 3.5 and we're using multi 5, right? So fine tuning. So is using the Hugging Face. And it turns out, so the library we generated, the performance, the affinity is very good. We're using the molecular docking to test. So how good is the affinity? So it's much better. So multi 5 is much better than GPT 3.5. It's better than ChemDive. So also the hit rate is higher. I mean hit rate. So in the cutoff with the hit rate we generated using the ChemDive. ChemDive is very so they're very popular, like the diversity screening library. So yeah, we use that as a reference. And also some of the enemy toxic property also is better. Have a better bioavailability and better the blood-brain barrier, the causing BBB the properties. All right, so yeah, that's the and also we done like using the AI for to predict, right, to predict the photocatalysis, the reaction for the, yeah. So that one can be using for like the flow chemistry. It can be using for the drug molecule, the synthesis, right? So nowadays, so you can see so all the things. So all the things can be all the synthesis, all the multiple step synthesis can happen in just within one vehicle, within one like the reaction tank, right? So in that way, you have to just changing add the reagent, right, and get out the product, and also you can do apply it for different catalysts. So in that way, so we can build up the very good like the machine learning model, and we can monitor, right? Everything can be a very automatic things, right? We monitor the reaction, and whenever there's some change, right? So we can use in the machine learning model to just to predict, right? So what is the best catalyst? What is the best reagent to add? Right, so this kind of thing actually is the computer-based to assist the synthesis. It developed a long time ago. It's since like 1950, right? Since 1950 original. They have been using they're using these things for retrosynthesis, the retrosynthetic the analysis, right? Especially for natural product synthesis, but for photosynthesis, the photochemical the synthesis. So so mainly, right? So when people want to find out what is the best reaction, the mechanism, right? What is the best catalyst? So people just do trial and error. But now we're using we're collecting a very large data set for photochemistry. Very large for the photo reaction. Very large data set is overall we have collecting the overall the 11K reaction types, right? So we build out these things. So it can be using to predict predict the yield, reaction yield. It can be predict what kind of the catalyst, especially for the metal. For the photosynthesis, especially as we're using the metal as a catalyst. You know, it can be predict for like the reagent. So whether it's a base or it's an acid, right? So what is the concentration? So yeah, all the things we can use that. So and then we validate that. It can validate with the literature. The model building process is good. Actually, there's some reaction is not covered in the original database, and we're using this model, right, to predict that. It performs so well. So you can see, so this one, it is the literature data, right? It's not covered in the original data set. And then this is our prediction. It's very consistent. It's very consistent. And also, and besides that, and also we identify some of the new reaction type. new reaction types. So we predict the new catalyst is different from the old catalyst. It still works. We predict that works, and we get a very good yield. We got a very good yield. So yeah, so that's it. And also using this kind of things, since we predict the new catalyst, right? And then then we can derive the new reaction mechanism out of the catalyst, right? So it's for CC bond formations. It can have a very good like this cyclation reaction happen. So yeah, we do this kind of validations. Yes, validations. Yeah. So that's pretty much the work. Yes, for AIDD. But besides the small molecule, so my lab also we do a lot of like the ADC design too. ADC, well, now we know that so ADC now is very like the very forefront in the cancer therapy, right? So the AIDD definitely, so it's not only for a small molecule drug discovery, also it's very good for large molecule like the ADC or antibody based. So yeah, so this is a very recent publication. So we're using the AIDD just for to test the size of the pack. Also, we have another publication is about we test what is linker, right? So we're using to test is what kind of linker work the best, right? So for ADC designs. Yeah. So yeah. So so I'll say now. So I think that the take-home message is so. The AIDD can really work. It really works. So and nowadays, so all these kind of like AlphaFold or like the NVIDIA, they have the BioNema. So this kind of platform is now is very accessible, very available. So you can use you can go to the Google Lab, right, or using the cloud computing. They all have the available. So it's very good for just for researcher. I mean, I really encourage the different field of the researcher just to use it, right? Use it since it works so well. All right, yeah. So that's pretty much that's it I want to share. So I'll be happy to take any questions you have.
I have a question.
Go ahead.
Okay, yeah, yes.
So, I guess when I read about AI, the way that it's used, including AlphaFold,
Okay.
Is that it takes things that are known,
Yeah.
like structures,
Right.
and then it computationally works those things over to find something new, right? Or to solve a problem.
Right.
But I kind of like getting back to Trent's point. What about the things that we don't have structures for and we don't know, right? But is new knowledge in drug discovery. So I guess what I'm saying is,
okay,
like, are we just taking the information that human beings have acquired over a couple of centuries in chemistry and categorizing it in very useful, powerful ways, but not really uncovering new areas of chemistry and how they might solve disease problems in human beings? You understand what I'm asking?
No, yeah. I think the AlphaFold, right? AlphaFold, the 2 or 3, right? It came out quite a while. I mean, there's a big discussion about the applicability, right? Or AlphaFold 2, especially when 2 came out. So yeah, so I'll say. So yes, the machine learning definitely they have we normally have the training side, right? Have the training side. So the some of the prediction is, but it's not always the case. So I think it just depends on the structure. But if the structure is very regular, just like the like a very regular alpha the alpha helix or the beta shape, right? So this kind of thing is not is conventional. Yeah, even though you got a new species, right? But the structured element is very conserved. It can still predict so well. But if it is very unique, like a second structure, nobody have seen before, I mean, that created a problem, right? Yeah, but I would say that, but the model, I mean, for machine learning and AI, so we call that this kind of problem is applicability domain. So whenever we make the prediction, we always test. So that prediction, if that prediction is within or without the applicability domain, we always ask that. But if it is within, we predict with a higher confidence. Without, so we're a little bit conserved. Yeah, but I can see the case I showed that for the photosynthesis, right? We identified some of the very brand new the catalyst. It works. It never been reported. Yeah, and also we derived the new reaction mechanism out of that. Yeah. Okay. Okay.
All right. I apologize. We ran a little bit over time, but I would love to thank our amazing speakers again, Trent, Simon, Abhishek. Thank you so much for your great talks. Also, I would love to thank our co-organizers, Frederick County Chamber of Commerce, and the big shout out goes to Burgess and NIPO. Thank you, Ryan, for being here, for breakfast, sponsoring our breakfast today. So thank you so much. And I just wanted to quickly say that at the end of May, we're going to have our next Biotech Connector event, which will be focusing on organoids. And Matt, who was just here from Thermo Fisher, so will be one, and Nathan, and Paul from Mimetas. So if you can join, please join us, come again. And last but not least, we ask all our guests to, grab some coffee and bagels and, use the restroom and proceed to the exit, because we do have some visitor rules in the building. So, but thank you so much for coming. We really appreciate that. Thank you. Hope to see you soon.